Jeg ved ikke, hvem der har lagt den jakke der, så goddag. Jeg er lige blevet forberedt til en dag, så jeg tænker, at jeg er rød i
doing one of the programs I carried in for whole calculator stuff. Get my second degree. I see. Oh my god. I wonder if it's part of like my team. Nerd. Ah. Cool. These are tired. Like an exotic country. Like I know those you will find someone.
Hi and welcome. First of all, this will be completely in English because we have a large audience who is live streaming this all around the world from other iGEM teams. You will hear more about iGEM in a moment. We're just waiting a little bit. We're a little bit late because we need one lecture. But please take some coffee, some snacks, water, and for the people live streaming this, I'm very sorry. We will be back in hopefully 5 minutes. Thank you. Okay, I think we're ready for this show to roll. Welcome everybody to the iGEM SDU 2019 Teams Antimicrobial Resistance Symposium, which is a very long title I just came up with. Today we will be having 4 different speakers and the iGEM team talk with different perspectives on antimicrobial resistance, which is a problem, as you all might already know. And we will be taking pictures throughout the day, so I hope you did your hair and look your best. The whole thing will also be live streamed, but from the back, so you won't be able to see your faces there. You're welcome to go up and take some coffee and such if you need it. And toilets are down the hallway there or down the hallway there by the end. So yeah, the first presentation will be by the iGEM team themselves, where we will talk about what iGEM is and what we're working on this year. In between all the talks, there will be a break, and after State and Serum Institute and Ude has talked, there will be a 20 minutes break where you can go ask the iGEM team or Ude or some of the other speakers about questions and take some more coffee, because we have 15 liters we need to be drinking by the end. So yeah, welcome.
There we go. Okay. Welcome. My name is Emma. I'm from the SDU iGEM team of 2019. We're here today to talk to you about our project, which we have named Conjugate. It's about antibiotic resistance, trying to find a solution to this huge problem. And Annemette will talk to you about that a bit later. But firstly, who are we? We are a team of 11 students. We're from 2 different faculties at SDU. The Faculty of Science and the Faculty of the Technical Faculty of SDU, from 4 different studies, and we've been working on this project, this huge project, since December last year. So we're very excited to talk to you all today about what we've been doing. The reason that we're hosting this symposium is because one of the requirements for this iGEM competition is to reach beyond the laboratory, to talk to you guys about how our project can affect the society that we live in, and how society can affect our project. And first of all, the iGEM competition. If you've never heard about this before, it stands for Internationally Genetically Engineered Machine. So it's a competition that 350-ish teams from all over the world participate in each year, and we all have to create a project that focuses on synthetic biology. So synthetic biology is a discipline, as the name implies, that is about taking a natural occurring biological system and engineering it genetically so that it can do something that it didn't do originally. So a nice example of this is the production of insulin. This is a result of genetic engineering of yeast. So this is what each team does, something within synthetic biology, and we all meet up in Boston in USA at the end of October, where we present our project to a lot of judges, and then they evaluate us, and maybe we win a prize. We hope. As I said, we do a lot of things outside of the lab, if this works. No? There we go. So, of course, we work in the laboratory. We try to make this system work, but besides this, we're doing a lot of social and societal projects. The biggest one of those is our ebook we've been writing and designing for the whole summer. It's a project that we're doing with a local high school here in Odense. We have been given 8 hours of their education in biotechnology, and we have to teach them about antibiotics, microbiology. Very exciting. It actually begun this Monday. Super exciting. We've been doing many different workshops and talks with other high school classes all over the country, and then last but not least, we've been very focused on trying to convey a message to the Danish society, both citizens but also decision makers and people with power, that we believe that synthetic biology has the potential to solve a lot of problems in our world. So this is Margrethe Vestager, who you might know. But that is also the reason for us hosting this event today, to try to reach out to people who might not know what synthetic biology can do. So what is our project about?
Okay, so first of all, I'd like to briefly tell you what antibiotic resistant is, if any of you have any doubts about it. So normally a bacteria has a lot of different mechanisms for it to survive, for it to proliferate. For example, it can create a cell wall and has a lot of vital mechanisms. An antibiotic is a drug, it's a compound that goes into this bacteria and inhibits some of these mechanisms. For example, penicillin is a compound that can go in and block the making of the cell wall, and this causes the cell to die. So antibiotic resistance is when the cell finds a way around this. It finds an alternative pathway, or maybe it blocks the antibiotic, or it straight up pushes it out of the cell again, which means that in the end it will survive, and we cannot use the antibiotic to kill it. So why is this a problem? Well, today there's the big killer is cancer, with 8.2 million people dying from it. There's 1.5 million people dying from diabetes, and right now 700,000 people are dying from antibiotic-resistant bacteria. But there's this O'Neill report from 2016 that states that in 2050, 10 million people will die from antibiotic-resistant bacteria. So this has the potential to be a huge problem. So what can we do about it? Well, back in January, we got this idea that we wanted to find some kind of solution. So we chose to focus on one specific bacteria, the E. coli that is in your guts, that has one specific resistance called OXA48. And we wanted to try to target this bacteria. So the way we want to do it is that we have created, or we are trying to create, a synthetic E. coli that has a specific plasmid, which is a specific circular DNA that we have designed. So this plasmid encodes conjugation genes, the Cas9 protein, and single-guide RNA. So what is this? Well, the conjugation genes has this effect that it can make our bacteria conjugate with the resistant bacteria. And conjugation is when the two bacteria create some kind of bridge between them so they can exchange material. So in this case, our bacteria will give our designed plasmid to the resistant bacteria. So when we have delivered our plasmid, the idea is that it is going to express the single-guide RNA and the Cas9 protein. So the single-guide RNA is designed so it finds a specific sequence in the gene, which is the resistant gene, the OXA48 gene, and then combined with the Cas9 protein, they can find the gene and cut it, thereby destroying the gene so it does not longer work. So the bacteria that was once resistant is now sensitive to antibiotic. And this has the potential to be a preventive treatment. So if you come home from a vacation and has become a barrier of this bacteria, this could be a treatment where you are treated and then the bacteria is no longer resistant. It can also be a treatment for when you are sick, you have become sick with this bacteria, so you first get treated and they become sensitive to antibiotics, and then you can treat with antibiotics afterwards. It also has the potential to target many different resistant genes because you can just change the single-guide RNA. So this is our idea of, it's just one solution to the problem with antibiotic resistance. So yeah, does any of you have any questions for us? Okay, so the question is how far we have come with our project. So not that far, actually, unfortunately. We are still working on our proof of concept, which is we have designed the plasmid, And now we are testing in the lab if it works on fluorescent proteins, if it can downregulate those. So hopefully in the next couple of weeks we will have made this proof of concept. So the question is how we can avoid the bacteria conjugating with other non-target bacteria. Well, first of all, conjugation is a very natural process that happens all the time. And second of all, our guide RNA is designed so it only targets a very specific sequence in the genome. So even if it conjugates with other bacteria, it shouldn't really have any implication in them because it should not cut any of their genes.
So just a short break where we will get Ude's PowerPoint on the slideshow, and then we'll be back in a few minutes. Hello.
So like how do you know which one to target? You'll say there's nothing goes on, and then you can just do it standard. There must be less most common mutations. So that's not
Okay, we're ready. So our first external speaker is Ude Wolf-Sørensen, who is a senior physician and clinical microbiologist at the reference laboratory for antibiotic resistance at Statens Serum Institute here in Denmark, who will be talking about status quo in Denmark, as far as I know.
That's right. Thank you very much. Thank you for inviting me. I studied here in Odense some years ago, together with Ulrik. Yes, so my name is Ute Sønksen. I've been working with surveillance of resistance for about 5 years. Before that, I worked mainly with antimicrobial stewardship. And before that, I worked a lot with taxonomy because I'm a clinical microbiologist, just as Jørgen and Ulrik also are. So, and I'm very happy for the slides you chose, because mine kind of follow what you already started. I will talk a bit about what is the situation in the world, and then I will talk a bit about what's the situation in Denmark, and we only have like 25 minutes, so I try to do it short, but you can ask questions, and maybe I can get you some more slides if you're very interested. No? So this is a map of the world, and we don't really know. This is made by a collaboration between a university in Washington, D.C. and an Indian research institute, and what they do is they collect data from surveillance systems all over the world, and they combine them in the map. This is a map showing Escherichia coli, E. coli, being fluoroquinolone resistant, and then the color follows the resistance gradients. You can see up here, if it's very dark blue, it's close to 100%, and if it's very light blue, it's 0. And it shows you that from many, many countries in the world, we actually don't have data, which is very troublesome, worrying, because we fear that probably the resistance rates are very, very high, and nobody really knows about it. The other thing that is difficult about a map like this is looking at some countries. This is Russia, and it's dark blue, as you can see. I think the average rate in resistance here was 78% in invasive E. coli. But this number is only based on 52 samples taken. If you go to Europe, you get some very good data, and the further north you go, the better it gets, and that is because we are in Denmark and Scandinavian and the northern European countries. We're very good at sampling our patients. You need to have samples to be able to do a surveillance. And we've been joined in the EARSnet program for about 15 years now. Some of the southern European countries have also been in it for years. They are getting close to nationwide observations now. The Eastern European countries are a bit lagging behind, but it's getting better and better. So if you want to have really good qualified data, you should go and look at Europe. But then, of course, Europe also has a lot of initiatives, while the rest of the world probably doesn't. So these are the main causalities for antibiotic resistance. There's increasing wealth. Low and middle income countries in the world continue to increase their use of antimicrobials. That is because they have them available by now. They didn't have to. I mean, they didn't have before. They will probably surpassing high income countries by 2030. That's a big issue. There's a tradition of using the most effective, and I have called it the doctor's pride. Maybe you should call it something different, I don't know. But in general, if you are a medical doctor and you actually have antibiotics available, then you want to treat and cure your patient. That is the most important thing to you. So in many countries of the world, the medical doctor does not think about that he or she will create resistance simultaneously while treating the patient. So there's lack of knowledge among doctors. Then there's the availability of drugs. Is the most appropriate drug actually available? In many countries it's not. Penicillins are not available in African countries and Southern American countries. So who decides which drug is on the market? And how is the market driven? And are drugs handed to clinicians for free, or are they handed out through the industry? And then there are the expectations and demands, and if you go and look at studies and publications from the UK and Scotland, you will find that they've been very interested in this specific topic, because they think that if you as a patient go to your doctor and you demand to be treated, then the doctor will hand out antibiotics. Then there's the whole laboratory and surveillance side, which insufficient or non-existing laboratory capacity. We have tried to join Kenya. They try to be part of the GLASS surveillance program, which is a program that was initiated by WHO. And their laboratory capacity was a disaster. And when we started talking to them, then their main focus was to start ensuring that there was actually something generating energy enough so they could have heating and cooling rooms, because else they would not be able to culture things. Then there's the surveillance, which is non-existing in many places. There is no infection prevention control. There's lack of knowledge regarding the principles. There's no reporting. So even if they take samples, which is quite common in the old Soviet Union countries, then they don't necessarily report the results back to their clinicians. So then the clinician will not know what to treat. There's no screening of risk populations. That does not happen very much in the Scandinavian countries either. It's increasing, but still not a lot. There's no sharing of knowledge and data among professionals, and there's no understanding of the public health related to AMR. So I'm very happy that you're trying to work with high school, which is great. And then what is the evidence? This is misuse and overuse of antibiotics in animals and in humans. The animals are the lighter purple ones, the humans are the darker purple ones. There is a high contribution and there is high evidence that this actually matters. But for everything else, more or less it is speculations. We don't necessarily know whether the factors that we are worried about contribute to resistance or not, because the evidence is not existing. But we can have theoretical expectations regarding that. So if we are in Denmark, like today, we think that travel probably is a very high risk, not necessarily for creating resistance, but introducing it into a population. And in Denmark, that would be primarily at hospitals. And once they're introduced to hospitals, then they can spread and create problems. In many countries in the world, diagnostics is a very, very big problem. Probably it does not really contribute to creating resistance, but definitely it could contribute to reducing antimicrobial consumption, and then that could reduce the occurrence of resistance. So that is also good, because that's what you're doing, right? Trying to do something, not necessarily that is diagnostic, but that can treat rapid and fast. This is a very old slide. I like it very much. It states that antibiotic use is the main driver for antibiotic resistance, but there are many factors. Above the arrow, you have all the human factors, the behavioral factors. Underneath the arrow, you have all the biological factors, which also would be antibiotic residues. That has been a big issue in many European countries. Cross-selection of resistance, you treat with one antibiotic, but then you actually select for resistance existing towards another antibiotic, gene transfer, non-antibiotic selection, and the dose and duration of the treatment. And up here you have everything else. How do I use it? Do I have infection control? Do I move my patients around? That's a very big issue in Denmark right now, because patients are moved constantly between the hospitals. Do people travel? Do food travel? What are the socioeconomic factors? And then, as I talked about before, culture and tradition, maybe corruption also. If money goes into a country to help build up a surveillance system, but it goes straight into the pockets of someone, then it will not do any change. So this is the strategy that was made by WHO in 2015, and it states you should have awareness creation, knowledge building among the public. You should do more sampling, more diagnostics, and more surveillance. You should reduce the incidence of infection through sanitation, hygiene, and infection prevention. You should optimize the use of medicines, and you should try to develop economic cases where the investment should go, and that should go maybe into diagnostic tools, maybe into vaccines, maybe into something else. That was followed by a list made by the WHO that stated now which bacteria or species and resistance combinations are the ones that we are really worried about and that we should start to survey. And they divided it into 3 areas: the critical ones, which primarily cause disease and problems at hospitals; the high priority ones, which are more in the community, although this one probably should move up to the critical ones; and the medium ones, which also are in the community. And then we made our own list. So these are the big 5 in Denmark. It's MRSA, it's VRE, methicillin-resistant Staph aureus, vancomycin-resistant enterococci, ESBL, extended spectrum beta-lactamase and gram-negatives, CPE, which is kind of belongs to this group as well. It's carbapenemus resistant, and the Clostridium difficile, which are not necessarily resistant, but they participate in creating a certain pressure of antibiotics consumed at hospitals. And we made a national strategy. We had one from 2010, which was more overarching, more unspecific, and then in 2017 we had a One Health strategy, and we also developed a national action plan for antibiotics in humans. And it states that we should reduce overall consumption in primary care. Primary care, that's the general practitioners, but also other people prescribing antibiotics in the primary sector. We should keep penicillin V the most used antibiotic, and we should reduce antibiotics of critical interest at hospital levels. And then there were no specific targets on the reduction of resistant bacteria, which I think is a big problem. So this is the Danish model: ample quality assured diagnostics, surveillance of the results, infection prevention control. There is a central infection control unit at SSI, which has existed since 1974, and then we should keep on using antimicrobials more prudent. This is what I do. I am the editor of the Denmark report together with DTU Food. We look at consumption in animals, consumption in it should say humans. We are kind of animals as well. Resistance from animals in zoonosis and in indicator bacteria, and resistance in human isolates. And then we try to figure out, are there any relations that are going across? So if there's a high consumption of something in animals, will that have an impact on the isolates we find in humans, for example? And the small bubbles are the new ones that we are trying to introduce or include in the program. So that is VRE, it's CPO, it's bacteremias in general, it's MRSA from pigs. You probably have heard about livestock-associated MRSA. And then we have veterinary preparedness, which does not really belong to this, but it creates more collaboration between the human and the animal side, and that way maybe we can also work together more closely. So then there are bacteria where you can measure your consumption. That's the red curve, and it will, that's the pillows behind, and then it will increase, or it will follow and create resistance in that species. So here we have E. coli and ciprofloxacin resistance, and it's a slide from 2015, but if you continue at 16, 17, 18, then the consumption is going down, and so is the resistance. So here we have a direct link, and it's very easy to measure, and it's also very easy to handle, because if you just reduce your fluoroquinolone consumption, then you will probably also reduce your resistance. But in other bacteria, it's much more complicated. Yeah, it's jumping. So MRSA has been notifiable since 2006. What does it mean when it's notifiable? It means that you screen patients upon admission to hospitals. You ask them whether they have been traveling or whether they have been in a condition where they maybe could acquire an MRSA. You then isolate them, you sample them, you send the samples for investigation, and if they're positive, you know that you probably, hopefully, can eradicate the situation. 2006 and 2012, there was a revision that was made due to the livestock-associated MRSA that was increasing. And in 2016, another revision was made where the contact with live mink has become a risk factor. So today, it's hospital travel, it's going abroad, it's working within the animal industry, which primarily then is pigs and mink. And if you look at this, these are the epidemiological classifications of the MRSA that we get. Then you can see the purple ones, those are the ones we call hospital acquired. It looks pretty well, maybe because you can find them and eradicate them, not the patients, but the MRSA. But here the number of community acquired infections is increasing, so maybe we have to revise and find out what we can do about that. Carbapenemous producers. There is no slide from next year, from 2018 yet, but I can tell you that in 2018 there were 115 patients, and by now we know for 2019 that we will be having even more, so this curve keeps increasing like that. And you can see that the dark purple one is the type we call the NDM, or I'm sorry, the OXA48. This one is the NDM. So there are different types of CPO, and we try to characterize them when we get the samples, and then we try to follow how they spread to be able to help hospitals to control. And this is the last one that we are worried about. It's the vancomycin-resistant enterococci. Over here, you have enterococci that were found in blood cultures corresponding to bacteremias. The blue ones are Enterococcus faecalis. The green ones are Faecium. You can see that that one is definitely increasing. And this red curve that jumps up shows you the resistance in the Enterococcus faecium. So in 2017, we had 7.5% of all bacteremias with Enterococcus faecium that were vancomycin resistant. So then should we make VRE notifiable? Would that help? And how should it be made notifiable? Because then we need to know how is it spread, so we can point out what are the risk factors, and we can try to work on it. For the time being, we have all strains referred to SSI. It's on a voluntary basis, but it is nationwide. And then we do whole genome sequencing. Huh? Yes. So these are data from YesNet. We're back at E. coli. E. coli examined in resistance against aminopenicillins, fluoroquinolones, third generation cephalosporins, aminoglycosides, and carbapenems. And you can see if the strain is fully susceptible, it is blue. If it is resistant to one antibiotic, it is yellow, and then it becomes darker and darker. So the very dark ones is at least 4, I think. Yes, resistant to at least 4 antimicrobials. And here you have the European countries that participate in the surveillance. You can see that Finland and Iceland and Norway and Denmark are on top, so Scandinavia is very well situated, the Netherlands, and the further down you go in Europe, and the further east you go in Europe as well. Bulgaria has a very low percentage of fully susceptible E. coli. The rest is resistant in one or another way. And that becomes kind of a vicious circle, because if you have many resistant bacteria in your clinical patients, then you will need to take more broad-spectrum antibiotics to treat them. How are we performing in Denmark in the use of antimicrobials? These are historical data. They go back from 1978 until 1991. We used to get data from the industry reporting to us. And when I say us, it's actually the Danish Health Data Authority on their wholesales. Then the medicinal statistic register was developed, and then they started to do nationwide registration of consumption based on sales from the pharmacies upon prescriptions and from hospitals. So these are 100% coverage of the Danish population, but still kind of follows the same trend. This slide ends at 2012. You can see that the highest consumption ever in Denmark was in 2011. And since then it has decreased pretty much the last 2 years. If you look at it in a different way, then you can look at the number of prescriptions that are redeemed per 1,000 inhabitants, and then the curve, this curve is kind of follows, it would be up here, following the consumption in DID, peaking at 2011, and it has decreased as well. So in 2018, we were down to 450 prescriptions per 1,000 inhabitants, but our goal is by 2020 to be down at 380 prescriptions. The question is whether we will make it, but I think we will. At hospitals, things are much more complicated, because also here, even though we don't have that much resistance, we have an increasing number of patients that are positive. Maybe I should show this first. So these are bacteremias, E. coli at the very bottom. E. coli contributes with 50% to all the bacteremias. Then you have Staphylococcus aureus, Streptococcus pneumonia, Klebsiella, Enterococci, and Pseudomonas. And you can see how the number of bacteremias just keeps increasing. From 2010 to 2018, it has increased with 50%. Also, the numbers of blood cultures taken is increasing a lot, and the numbers of patients that are blood cultured. And we don't exactly know why, but we have an idea that probably it's due to changing demographics. Maybe it's due to more people being treated in more invasive ways, more catheters being used, more invasive treatments being used. And that will drive your consumption at hospitals. So these are the critically important antibiotics, and in Denmark we have defined that for us the critical important ones are cephalosporins, fluoroquinolones, and carbapenems. If you look into the rest of the world, then the group of critical important antibiotics is bigger. This is the percentage of the total use at hospitals. So you can see that in 2009, about 35%, this is the national average, 32% I think was the national average, of all antibiotics used at hospitals would be one of those three. And by 2018, we are down at 22%, but now things are not really changing that much anymore. And I think what needs to be done is that, especially within surgical procedures where a lot of cephalosporins are used, you need to consider whether you should change to other antibiotics if you want to bring down the cephalosporins. We're back at Europe. This is the consumption measured in all of Europe. The darker red it gets, the higher is the consumption. We are yellow. There is an overall assumption that if you pass 15 DDD, defined daily dosage per patient, you will maybe start creating resistance, but at least you will kind of contribute to pertaining it. And we are, I think, at pretty much 15 right now. So hopefully we will go below that. These are data from the animal side. We're back in the E. coli. We're looking at chicken. We're looking at ciprofloxacin and resistance, and you see the same trend. The lighter blue it gets, the further north you are in Europe, and the darker it gets, the further south and east you go. And it's the same system of dividing. This is full, susceptible, and then it becomes more and more resistant. This is also consumption. We are here. You can see that if you look at how much antibiotics are used for the treatment of animals, and you look at how much is used per animal, then we are performing pretty well. And I don't remember if I brought the other slide, I'm not sure. No, I didn't. But we have 36 million pigs that we produce in Denmark. So although we maybe only use a little bit in each pig, then still we use quite a lot. So every time you measure, you have to consider, what is it I'm looking for? What do I want to measure? Yeah. So what are our main lessons? We know that collaboration, that's what works. We know that it's very important to include different specialties and fields if you want to do a good surveillance. It is important to speak with one voice. I didn't really talk that much about it, but if you want to have political awareness, then you need to use your common voice and not one saying one thing, another one saying a different thing. It works if you have a bottom-up approach. Why did I write that? That's because the whole thing on surveillance started on a voluntary basis and as a research project, and then it kind of came into political awareness, and they started financing it on a more regular basis, and I think that's very important. You can start up with bottom-ups, but you need governmental support and structure and funding if you really want to do a change. You need to define your short-term goals and your long-term goals. Short-term should be something where you have success, so everybody is happy and wants to move on and feels like it's encouraging. And then on a long-term basis, you need to establish systems that are sustainable. And what are we good at in Denmark? We are very good in taking samples. That is a bit threatened right now because there is so much from the industry coming in with diagnostic tools that are delivered directly to the clinical departments. And if they take their samples themselves and they do their analysis themselves, which is no problem, we will not get the result. So we still will need to get to know what's actually going on there. We're good at doing analysis. We are very good at doing registers and databases. We have databases that go back to the 50s. And we're probably also very good in ensuring that results are given back so the clinician actually has something to treat upon. And then in general, we are very trustful and open-minded, and we will work together, which is very good. When I talk to these people from Kenya that I mentioned before, they are also very open-minded, but they are definitely not willing to work together, and then things become difficult. Here's a slide with some links if you're interested in more. And thank you.
Are there any questions?
Yes, we start in the... Oh, you go back, okay.
Thank you for the talk. You say that the main driver for antibiotic resistance is antibiotic usage. And in the map over the countries where they use the most antibiotics, Finland was not one of the countries using the least, yet they were still the country that had the least resistant strains. Do you know what they're doing to achieve those results?
I think you need to ask again, because I think you're saying that where they not used a lot, they still had a lot of resistance. No, that's not what you're saying. You said the opposite.
You're saying the main driver
yes.
for resistance is usage. And Finland was one of the countries which did use, they weren't one of the countries that used the least.
No, that's
yet they are one of the country which had the least resistance. So I assume they do something else to achieve these low resistance rates. Do you know what these things are?
But they don't lose a lot of antibiotics. They don't use a lot of antibiotics. So on the map where I showed all the colors, they were not highly red. They were still yellow. So their consumption of antibiotics is close to the Danish one. They use less at hospitals. They don't survey as much as we do, so maybe they would find more resistance if they would look more for it. But in general, they are performing pretty much like the other Scandinavian countries. But the Finnish hospitals are very good at doing infection prevention and control. So that could also be a reason for not being as high in resistance as other countries are. But in general, if you put the maps close to each other, then the colors will follow each other.
Thank you.
it is coming to Europe. It is here. The carbapenemous producers that we have in Denmark, well, maybe one of them was made by ourselves, but most of them were introduced from somewhere else. But we still can control the spread of it. So if you find the ones that are introduced, you can prevent that they are being spread further, which is, I think, one of the most important things. The other thing I personally think is very important is that antibiotics today are produced primarily in Asia. There is no regulation on environmental factors that they should keep in mind. There are a lot of studies reporting upon, for example, India, where fluoroquinolones are produced, that the concentration of fluoroquinolones in the water, the wastewater that comes out from the factories, is higher than what you would need for the treatment. So you can just drink the water and then you did not take your pills. And if we, as in Europe and also in Denmark, would start to define and say that, yes, we want to buy your antibiotics, definitely, but we demand from you that you produce them under good conditions, and then we are hopefully also willing to pay more for them, maybe that could change things, because that in itself creates resistance in those countries. And that's just one tiny thing, but I think that could, contribute to controlling it somehow.
Okay. Well, thank you very much. We will now have a 15 minutes break, so we will be back at 1:25 with Doctors Without Borders. So yeah, take more coffee. It's free. Okay, in the meantime, while we get the technical parts to work, I would like to welcome Alexandra Kruse from Doctors Without Borders. She's a pediatrician specializing in pediatric infectious diseases, and she's going to talk about antimicrobial resistance, as you might have guessed, but in the Médecins Sans Frontières kind of perspective. So, take it away.
Now, so this is Otim. He's 4 years old. He has been exposed to TB. He is malnourished, as you can see. He's very lethargic and feverish and irritable. All signs of severe illness, and could be sepsis, so bloodstream infection. But we are in a hospital tent in South Sudan, in a refugee camp in South Sudan, and all what Ute was talking about surveillance, resistance, all of that, forget it. So maybe they only have 50 E. coli from all of the former Russia, but here you don't have anything at all. So what I know is that from the estimates of global child mortality, more than half is associated with malnutrition, and I know that infectious diseases are among the main killers. So here I am with Otim, wanting to save him, and then in the back of my mind having antibiotic stewardship, so how to use it rational. So this is a dilemma that we face every day working for MSF. We have more than 8 million patient consultations all over the world. We are there where people need the most, so it doesn't matter where that is, whether it's the Mediterranean or whether in Gaza. We are where the people are most in need, and we cover from basic vaccination campaigns to more complex surgery. So it's the whole spectrum. And besides offering medical aid, we also do témoignage, meaning speaking up of what we see. So we are testifying and bearing witness, and we are also doing research, and you could argue that that's a kind of medical témoignage. So this is one of the places where we face resistant infections. This is in Gaza. This is in trauma patients having complications with infections and resistant infections. And so Amir, this young guy, he didn't know he had an infection, but when he wakes up after surgery, he's dead. This is one of the places where we work. But we also work with other war-wounded patients from Syria, in Jordan, in burn patients in Haiti, and they are particularly vulnerable because of their barrier damage, like the child you see in the corner. We also work with newborns in Pakistan, where we also have resistance problems with Klebsiella as being one of the bacteria, and then of course multi-resistant TB, both in South Africa, India, and Eastern Europe. So these are some of the challenges we face. Globally, you already heard from Ute, but consumption is increasing, and so is resistance. We have new antibiotics in the pipeline, but limited, and now we face mortality of about 700,000 people dying because of resistant bugs, but already now we have more than 5 million people dying because they don't get the antibiotics that could save them. So it's not only a matter for us about resistance, but also access. So all the good interventions that you are going to create, remember to have in mind to make it accessible and affordable and implemented. Like we talked about, data in low and middle income country is lacking. So what do we face in the field? If you're lucky, you have this guy, but usually you don't. So as Ute already mentioned, disproportional burden of infectious diseases and resistance patients are vulnerable, like Otim that you saw before. He's malnourished, so he's more, he has less of immunity to fight an infection. They can also be vulnerable because they are refugees, they are on the move, so it can be difficult to comply with your treatment. It could be for TB, could be for HIV. You have human resource insufficiency, so either they are not there, or if they are there, maybe they are not paid, maybe they don't have updated education, maybe they don't have books. So insufficiency on several levels. Also, infrastructure and logistics can be difficult. So some of the tests that we use, it's easy because we have cold temperature, we have a fridge if we need that. In many of the settings we work, we don't have. We have limited diagnostic tools. Like yesterday, our microlab in where I'm working showed us a new test for dengue fever. That's a fast test to use, but it has to be on temperature that we can't use in the field. So in the field is where you have dengue and where you have the complications with the dengue. Here, if you have it in a tourist who has been out or traveler, usually you can be very fatigued, which is also annoying, but it's not life-threatening. But in the field it can be because of the children, particularly who are at risk and have it several times. They can have lung edema and hemorrhagic shock. So observations options are few. So if I see a child here in the emergency room with an ear infection, I can say watchful waiting and I can explain and please come back. If I'm in South Sudan, maybe they've been walking for one day, so it's difficult to say just walk back and then walk here again. So that is difficult, and also we have limited space in our hospital, so just to admit for observation is not easy. I can do that in my hospital here, but not in the field. Infection control and hygiene is also challenging. We have overload of patients, so having the space you want to have and upholding the hygiene is limited. We have limited treatment options, and we have over-the-counter issues. So you don't have, sometimes you don't have to go to a doctor to have a prescription. You can just go to not even your pharmacy, but the local market and buy a few TB drugs that we are very afraid to use, because if you use only a few, then you're more vulnerable to creating resistance. So there's a mismatch in need versus access. So what do we do about it? Well, we speak to people like you. We also spent most of the money we got from the Nobel Peace Prize to launch this access campaign to create access not only for the people that we see in the field, but also for other people, the people that we can't reach. And also because in the field we see a lot of needs that are not addressed, so we said, okay, what can we do about this? And actually, two of our wishes on the list of 2019 are about resistance. So this is about affordable TB drug for multi-resistant TB and antibiotics for superbugs. That's where you come in. And I will elaborate later, but what we want is not charity, we want change. So a global fund was launched, not the global fund, we already have a global fund for tuberculosis and AIDS and malaria, but this is a global fund for R&D, because what we know is that this is not an MSF fund. This is done by several UN bodies and EU and all the big stakeholders. But what we know is that AMR and emerging infectious diseases and neglected diseases have in common that they are public health priority, but maybe not a priority for life science or for the pharmaceuticals. So we need to speak in one voice, like Ute was talking about, not even in Denmark, but globally. So we shouldn't get fragmentized but reconcile, and we should make sure to actually have the funds allocated to this initiative, because sometimes we have big nice launch and we have nice speeches and nice champagnes and bubbles, and then afterwards the money doesn't really come, or it can take some time. So, as I said, we also do research, and here are some of the publications that we have contributed to, and one is about how to implement clinical bacteriology in the field, how to find out of all the febrile kids we see, how do we find out who needs the antibiotics, who are at risk of having a severe infection. So this was a big study in Tanzania with more than 3,000 children. And actually we both managed, of course, to have, it was a non-inferior study. I don't know how much aware you are of clinical studies, but anyways, this study showed that with this new algorithm that also has some paraclinical tools in it, you can lower your prescription of antibiotics. And some of the concern was that maybe, like Ute was saying, maybe some of the doctors still wanted to prescribe antibiotics, even though the test showed they didn't have to. But actually what we found was that if the doctors and the clinicians and the families understood what was going on, why they needed antibiotics or why they didn't need antibiotics, compliance wasn't difficult. But of course, if you only know you don't get anything, please go home. People don't understand. But if you explain why, and that it is a disease, but it will pass and your child is strong and so on, we actually managed in this study. And then we know that once you want to implement studies in larger scale, it can be still challenging. Then we had a look at a big cholera epidemic in Yemen to see whether the strain was susceptible for the antibiotics we usually use. We also evaluated a fast test to diagnose meningococcal in cerebrospinal fluid. Usually here in your lab, you would have a microbiologist do all the stain and looking in the microscope and everything, but in the field it can be difficult. So to have a quick test that you can perform yourself is very good. Unfortunately, it didn't perform that well because it needs to be prepared still. And we have also contributed to addressing the 33,000 children that are thought to have MDR TB, and where drugs are a major problem. It's a problem amongst adults, and it's even more of a problem in children. So these are some of the publications we have contributed to. Then we called for, or we advocated for this joint effort to try to make a research agenda that are prioritized from needs and not from pharmaceuticals wishes, and where are the money, where can you earn the money, but where are the patients, where are the needs. And this was actually launched from G20 and launched in 2017, and then I think it was launched in 18, and there they try to create this hub for R&D in resistance. And what we just want to emphasize is that it needs to be patient, needs driven all the way. And you can't drive it by business as usual, because pharmaceutical is driving the business as usual. And so we stress very much that it needs to be coherence with the principles and policies of the drugs or the tests being available, accessible, adapted, and affordable, either or. And this may seem very obvious, but once it gets into the machine, it can be difficult and you can forget. And if this is not in the back of our mind all the time, then it won't help the patients that need it. And so we also stress that there needs to be incentives because the profit is not in this market compared to other markets. And there has to be a governance voice of the developing countries. But we favor, like talked about before, research coordination, of course. And then ICARS, I was just talking to Ute about it. This is actually launched in Denmark, and this is a center to oversee creating the data that are not there now for resistance in low and middle income countries. And we just talked about it. It was launched a few years ago and everybody was very happy, but I'm not sure the money is there yet, but we can talk about it later. Or you can ask about it if you want to. So, Denmark is also contributing, because what we need is stewardship. So we use the antibiotics we have rational. There has to be an indication. We need to use as narrow spectrum as possible. We have to have one health in mind, that we and the animals are connected. So what we do with our livestock and pets will also influence us, as we've already seen. We need improved diagnostics that are available. We need infection prevention, control and hygiene, education of staff, and also health education of our children and other patients. We need surveillance both of infectious diseases, prescription habits and resistance, and we need the new antibiotics, because we believe that medicines is not a luxury, it's a right. And so what we commit to in MSF to help children like Otim is that we are trying to provide better diagnostics to the fields that we have, that we are educating our patients, our families, our what we call health education, and our staff, that we contribute to research and we do advocacy also with our campaign. So AMR is on the top of our agenda in MSF Denmark and also in MSF worldwide. And we hope that you will help us to help. And here in Odense, actually, you have a volunteer group, and one wearing a T-shirt up here in the back, so, and one in the back also, in the very far back. So if you're interested at all, let us know. Yeah, that's it. So I hope we can spend the rest of the time discussing, because I find it so much more interesting than just me talking. So what you want to know, or what you want to address, or what you want to ask about ICARS, or how Denmark can contribute? Okay, then I will start. What about ICARS? How far are we in this surveillance of resistance in low and middle income countries that Denmark is hosting?
Let me start. Yes, ICARS is trying to approach the whole question of AMR from a bit of a different perspective. There are many approaches already. There is research performed, there is WHO is doing a lot of things. I talked about the global surveillance system, but nobody has really been there mapping what do the individual countries actually need. Because if you leave Europe and you go further south, specifically on the southern hemisphere, then they need clean drinking water, they need so much more control, they need a better health infrastructure, they need so much else than only AMR. So at the beginning, ICARS will try to map the situation and help to point out where should you start, where should you focus. And how far is ICARS then? So far there is 4 people working on it. They are situated at the SSI, but that's just geographically. There is a person from the Ministry of Health, there's a person employed by the Ministry of Health, and there is a person that has been within animal health and welfare. And then we were very, very fortunate to have a secretary who had been working on a global scale with AMR before, but she just left, so we need a new person if someone wants to join. What they're trying to do is they're trying to establish the governance structure that ICARS needs before they can actually start working with ICARS itself and the solutions that are needed. So they're still in the very, very beginning. And I spoke to Robert yesterday. Robert is the project manager, and he says it's if you look at similar systems where people try to establish organizations and things like that, then the time frame is very, very short. They've only been working with it for 1.5 years, so they think they are doing quite well. But then again, they need to actually be established by October, so maybe they're not fast enough. They need financing. They have the money from the government, who says we will pay for the offices, for the premises, but there is so much more money needed because you need to invest money to get money. One of the big issues is that the World Bank will help finance it, but the World Bank is a bank. They don't give you money, they loan you money. And if you want to borrow money from someone, you need money yourself. And so right now they're trying to get people on board and establish the structure around it, and that's it.
So not easy, but in progress.
I have a question from the iGEM perspective of doing research, because I know there are some clinicians in here, but I also see a lot of biochemists, molecular biologists, and so on. We are mainly working at the university research level, and you talk a lot about accessibility. Of course, we want our system to be accessible in the end, but how can we, as researchers at universities, push the industry into making it more accessible, because we don't have the resources to make it accessible? What can we do?
Actually, you have a very active student organization called UAEM, and they are actually working consistently and quite professional, trying to make universities more professional, because what is often happening is that we do some of the basic research and then we hand it over to the pharmaceuticals doing the clinical trials, which are expensive and complicated, but maybe we should be better at securing, or at least transparent on, what do we get out of it when we hand it over? What are the what are the rules and what are the laws, and what can we do to have it in mind from the very beginning? Because you can say that what we do in universities are not 100%, but a lot of it comes from public money. So I think that we as a public also have a fair right to know what's happening to our money. But having a bright idea like you presented this morning and having it all the way working in the field, it's a long road. So there are no easy fixes. But if we join forces, maybe we can make it. I mean, that's the only way to do it.
Thank you very much for the talk. We will be switching slideshows and then progress to the last part of the this lecture series, which is mainly going to focus on MRSA, methicillin-resistant Staphylococcus aureus, and we have a local expert coming to talk for us. Yeah, Jakob.
Ja. Det er fint. Det må.
Okay. So I would like to welcome Hans-Jan Kolmers, who is a professor here at the Clinical Institute at the Center for Translational Microbiology, which he told me to mention. And he'll be talking about one of his very dear subjects, MRSA, and I'm looking forward to that.
Thank you. Yeah, well, we are building up a new center for translational microbiology because we think microbiology has to be applied. Applied microbiology is important. Also to do something good to the rest of the world. That's very important. Well, my issue today, or my topic today, is MRSA. And to understand what is MRSA, I think we have to start out to look into what is Staph aureus. Because MRSA is basically only a drug-resistant Staph aureus. And that's actually the whole explanation. So if you know a lot about Staph aureus, you'll also know a lot about MRSA. Staph aureus. Everybody here is familiar with Staph aureus. Every fourth of you will be colonized with Staph aureus in your nose, in the nostrils, from where you spread it to the skin. 25%, up to 50% are transient carriers. Silent carriers, it doesn't really matter because you have the organism, but you're not sick. But the point here is that every now and then, people may become sick from this bacterium, which is not only a colonizer, but also one of our most important pathogens, giving rise to skin and soft tissue infections, and even more severe infections, septicemia, osteomyelitis, etc. Now, these Staph aureus originally were susceptible to all antibiotics, even to penicillin. But very soon, in the 1950s, penicillin-resistant strains emerged. Today, 90% of all our Staph aureus strains are penicillin-resistant. Now, this is due to beta-lactamase. And these drugs, or these organisms, can be treated with beta-lactamase-resistant penicillins. We invented the first drug in 1961, methicillin, here. But today, most of us use oxacillin or related drugs, penicillinase-resistant penicillins for this kind of organisms. Now, MRSA, as I mentioned, is a drug-resistant Staph aureus. That's a short version. It's not more virulent, it's not more dangerous, but it's harder to treat, and that's our main concern. It's harder to treat because we don't have the antibiotics available. Those drugs that we have do not work so well as old-fashioned penicillins, and they are much more expensive. So that's the issue. In Denmark, about 2% of all our Staph aureus strains will be methicillin-resistant Staph aureus. Methicillin-resistant Staph aureus, the acronym MRSA, equivalent to about 25,000 persons. So about that size. The reason for MRSA is that strains have acquired genes, MAC-A and C genes, that encode an altered penicillin binding protein, which is the reason why old-fashioned penicillins, methicillin, doesn't really work any longer. Now, this is quite dramatic, a quite dramatic situation, because these altered penicillin binding proteins mediate resistance to virtually all beta-lactams. That means all penicillins, all cephalosporins, and all carbapenems. That's the drugs we use. So we can see this is really a truly drug-resistant organism. And on top of that, MRSA strains may even acquire resistance to a lot of other antibiotics, non-related antibiotics, so that we are really close to a situation where we don't have very many drugs left to treat. So that's the issue, that's the challenge. The geography, we have already seen beautiful maps showing that Scandinavia, Northern Europe, is a green area where we have a low frequency of drug resistance, also of MRSA. This is the greener, the better. But you can see we are surrounded by red countries and yellow countries. So we may say that in our part of the world we're privileged. We are kind of oasis in an ocean of, or no, in a desert of resistance. To the south, Spain, to the east, Eastern European countries, and so on. The history of MRSA is quite fascinating. Actually, we have had 4 pandemic waves of MRSA since the first strains emerged very soon after the introduction of methicillin. Now remember, MRSA is methicillin-resistant Staph aureus, but you could also say it is multi-resistant Staph aureus, because it is a multi-resistant strain. But very soon after the introduction of methicillin, the first MRSA strains emerged. And really, this is quite, it has been shown that it was a quite amazing story, because it was due to one genetic event giving rise to what we call the archaic clone, which spread all over the world in hospitals in the 1960s, 70s, giving rise to surgical wound and device-associated infections. In the 1980s, things became a little more blurred because more clones popped up, but still there was only very few clones of MRSA spreading in the world. Still in hospitals, giving rise to surgical wound infections and device-associated infections. But then from the 1990s, something quite dramatic appeared. Now, and this was really new, now a new wave came up, and this wave came from the community. It was due to the spread of MRSA in the community, not in hospitals, but in the community. Apparently, MRSA became able, these strains were able to spread in the community, even where you have a low consumption of antibiotics. At the same time, a lot of different clones popped up. And the reason is that the MMEC gene became motile. It could be transferred horizontally from one cell to another. And this gave rise to a patchwork of many different clones. At the same time, strains emerged that were able to cause primary wound and soft tissue infections in people with apparently intact skin. We are still in this wave, but from the 2000s, something new emerged, and that was the pig-associated or livestock-associated MRSA that's coming up now. Still, it's a few clones, and they give rise to infections in humans, emerging infections. That's why we are concerned with these pig farms being colonized, pigs being colonized with MRSA. If they didn't spread to humans, nobody would care, but unfortunately they do. Now a little more about history, Danish history on MRSA. As you saw, the archaic clone that emerged in the beginning of the 60s hit Denmark quite badly. This is a blue line here, is the first wave of MRSA that popped up in Denmark. Denmark actually was one of the countries that was most severely hit. In the middle of the 60s, more than 30% of our Staph aureus strains were MRSA strains in hospitals. That's really something. It's a world record. And at the same time, these strains were resistant to a lot of other drugs, tetracycline and macrolides. Tetracycline emerged earlier than methicillin resistance, indicating that tetracycline probably played a key role in the selection for MRSA. Now, we eventually managed to control this epidemic by 3 major points. As Alexandra also mentioned, reduction of antibiotics, in particular of tetracyclines, improvement of hospital hygiene, and better microbiological diagnostics. That were the 3 key points, the key strategies that enabled us to control MRSA. And then you can see over the 80s and 90s, we had a world record of low prevalence of MRSA. It was not very easy to write articles from Denmark to medical journals because nobody believed us and nobody thought it was very sexy to talk about susceptible Staph Everybody talked about resistant strains. We were only presenting with susceptible strains, and that was not very sexy at all. But things changed, and as we have seen, this Ute also showed this slide. You can see it from the early 2000s. The new wave of community-acquired MRSA began also to hit in the Danish society. It's still not a very high prevalence, but you can see the trend is quite remarkable. We are in the beginning of a new epidemic, and actually this epidemic is out of control. You can see here, it's driven by the community-acquired strains that spread in the community outside hospitals. It's also driven by import of strains by tourism, for instance, or refugees coming to Denmark. And the third important factor is this livestock-associated MRSA CC398 that pops up now and becomes a factor of concern. But you also can see that in contrast to what we should believe, the spread of MRSA in hospitals is quite low. It's actually not really a problem. We have very few cases each year, actually, of spread of MRSA in hospitals. It is a community problem today. That's the modern epidemiology of MRSA. If you are in an area like the region of southern Denmark, where we have a lot of livestock, you can see that this MRSA CC398 will dominate the picture. 50% of our MRSA strains that we isolate in this hospital, in this region, are actually livestock associated. They have their origin in livestock. It's quite remarkable. And these strains, they're strains from humans presenting to hospitals with problems or being screened for MRSA on admission to hospitals. What's happening with these livestock-associated MRSAs is quite remarkable. The history has been unraveled, and actually analysis have shown that it started out with a susceptible Staph aureus CC398 strain being transmitted to animals. So humans transmitted a susceptible Staph aureus strain to animals. In animals, a number of events took place, loss of genes, enabling these strains to become accommodated to livestock, particularly to pigs. And then you know that in modern industrialized pig production, you use a lot of antibiotics, you use a lot of tetracycline, and you lose a lot of zinc, heavy metal zinc. And actually, due to this high consumption of these kind of drugs, MSSA became MRSA. It started out being tetracycline resistant, we know that. It started out becoming tetracycline resistant, and then MRSA came on top of it. And now these strains are transmitted back from livestock to humans and even to hospitals where they give rise to problems. So you can see this is really an example of what happens with resistance jumping from one ecological niche to another. We transmitted a susceptible strain to animals. Animals were treated with antibiotics, and the result was a retransmission to humans of a drug-resistant organism. You can see here, it's quite a remarkable amount of antibiotics being used in these different ecological niches. In animal production in Denmark, you use actually twice as much antibiotic measured in tons as you use in the whole healthcare system. It's quite interesting. You may ask, how can tetracycline or zinc become a factor of importance for the selection of MRSA? To understand that, you have to consider that genes are coupled. It could be on plasmids or similar genetic elements. And so if you actually use tetracycline, you will not only select for tetracycline-resistant strains, you will select for strains that have the whole package of resistance characters in this particular genetic element. So this is a reason, call it co-selection. So this is a reason why you can actually, you have to, if you look for the reason for MRSA spreading, you don't have to look into the consumption of methicillin, you have to look into the consumption of different drugs, even non-antibiotics, like for instance zinc. Why are we concerned? Well, first of all, because these strains, these MRSA strains, are hard to treat. And because they're hard to treat, because they're difficult to treat, we don't maybe not be precise enough to kill the organisms in the first round. Things take time, and maybe the drugs we use are not as efficient as old-fashioned penicillins. As a result, the mortality will be higher if you have, for instance, septicemia with these type of strains. You can see it's about, the mortality will be in the range of about twice as high as with susceptible Staph aureus strains. So it has a consequence for the patient. There's also an economic consequence for society. If you look at penicillin here, let's say penicillin is factor 1, you can see that vancomycin, which is the ordinary drug we use for MRSA, is 10 to 15 times more expensive than ordinary penicillin. And you go to a more modern drug, linezolid, for instance, you may be in the range of 50 times the price for penicillin. So if all Staph aureus strains became MRSA, you can see this would have a dramatic impact on health economics. So this is one of the other reason why we are concerned. What about strategies? We already talked about strategies. There's basically 3 main strategies to control MRSA and other drug-resistant organisms. You can stop selection by reducing antibiotic consumption. That's very important. But it's also very, very hard. And it's very hard in areas where you don't have access to diagnostics. You just have to treat patients in blind without really knowing what is going on. So this is really a challenge to reduce the consumption, but it's important because this will stop the selection of resistant strains. No antibiotics, no resistance. But you can also, the other strategy is you can stop transmission between patients or from the environment to patients by improving hygiene. So it's not only a question of reduction of antibiotics, it's also a question of better hygiene, better hospital hygiene, better hygiene in society. Hygiene is important to control drug resistance, but you have to see it in context with reduction of antibiotics. And then as a perspective, you have also to work to find new antimicrobial drugs, which Jan is going to talk about in a short while. The National Plan for Reduction of Antibiotics in Denmark, we have already mentioned it, Ute has mentioned it. In general practice, we have to reduce our prescription, our antibiotics. With antibiotic prescriptions should be reduced by 25% over a few years. And we are actually successful. It seems to work. We are very well off in this context. So this seems to work. And we should increase the amount of narrow-spectrum penicillin. That's a bit harder. We haven't really come up with more narrow-spectrum penicillin as compared to the other drugs. That's harder to achieve, but maybe we will find out. In hospitals, we have to reduce the consumption of what we call critically important antibiotics by 10%. That's the fluoroquinolones, cephalosporins, and carbapenems. And the veterinary sector also has to contribute. We have to learn to raise farm animals with less antimicrobials, in particular tetracyclines and zinc. Now, it has been quite hard to make people believe that tetracyclines play a role. But I'm so old that I remember the first epidemic wave of MRSA that you knew. I was a young doctor in the 1970s when we still had MRSA from the first archaic clone. And we used tetracyclines in hospitals and in society, just like the way they do it in agriculture today. It was the drug, the house wine, just for everybody. And this was really, so we really saw the result of restricting tetracycline. We almost abolished to use tetracycline, and it worked. So I'm a firm believer in that tetracycline plays a role, and we have to be quite firm on that. But also, we have to reduce the zinc because these strains in agriculture, they are actually zinc resistant. So this is an MRSA strain that has just found its ecological niche where you use a lot of tetracycline and a lot of zinc. That's very fine. It thrives there. It's just the way it wants to have it. What about hygiene? 3 important aspects of hygiene, which are not very sexy, I admit, because you have heard a lot about it and you say, oh my goodness, I'm so bored with hygiene. They all say, wash your hands, get a clean uniform, and get cleaning of the environment. It's not a very sexy message. It's a very basic message. Nevertheless, these are the key factors in controlling also drug-resistant organisms. So improving better basic hygiene in hospitals and in the community, in all kind of settings. That's very, very important. And then, as my last slide, what do we in what is our strategy in hospitals? We have a guidance for preventing of spread of MRSA issued by the National Board of Health, as we have heard. The strategy is a search and destroy strategy, which means that we screen for MRSA in risk groups of patients being admitted. We isolate patients and we destroy, that is, we don't destroy patients. We eradicate MRSA carriers as soon as we can, and then we follow up with controls. So actually this is not as in a hospital as looking basically in a very narrow perspective at hospitals. This is not a search and destroy policy. It's a search and contain policy, because you cannot eradicate MRSA from patients being admitted just for a few days in hospitals. That's not possible. So the consequence is, when we screen patients in risk groups and find them positive for MRSA, they'll go in isolation for the rest of their admittance to hospital. I think that was my last slide. Thank you very much. Thank you for your attention. And now, any questions? No? Alexandra.
I don't know if it's true, but I saw a funny Australian study about, and a French one, about how often doctors do or don't change their white coat. And of course the French were lazy and did it every now and then, and the Australians were the tidy ones. But actually they couldn't prove anything on any hygiene outcome and resistance and nosocomial infections.
Probably because compliance was so low that nobody changed their uniforms every day. So
the Australians said they did. But have you seen any studies? Just because, you know, every morning we were waiting in line for...
You're right. I haven't seen studies where you have made a control study where you did nothing else but changing uniforms and asking doctors to go with dirty uniforms in a control group. You wouldn't be able to make that type of studies. They will be, for ethical reasons, it would be impossible. This is actually our dilemma. You cannot actually pick out one single factor and test it. It's impossible in a hygiene setting. You cannot also really prove that hand hygiene works, because would you make a control group? Who would like to go into the control group being treated by doctors who didn't wash their hands for one single time during a whole working day? Nobody would. So that's the point. We have to look into this as a package. We cannot really single out the single elements in this hygiene package. It's impossible. We have to believe that this is what we do. There are plenty of Staph aureus on doctors' uniforms. There have been wonderful British studies of contaminated uniforms, and they actually made a competition in the British Medical Journal where they asked for ideas what to do with these uniforms, because they wanted actually to do away with uniforms altogether. It would be better people just to have their private clothes. And they ended up with, say, give them to the vets.
I have a comment, not a question.
Yes.
Maybe it's two comments. I've been visiting other countries to help them establish better systems in surveillance and in keeping control of antibiotic resistance. And when you go to the Eastern European countries, who used to be Soviet Union countries, then the whole way the hospital system is built is very different from the Danish one. And it starts with that you enter the hospital and then you leave your coat. And if your boots are very dirty, they will supply you with clean shoes or something to put over your shoes. And they will follow you all the way up to the department or the ward. So, and then once you come to the ward, there's only one single patient in a room that was built for, I don't know, 10 patients probably. So you don't have any overcrowding. And I think that's one of the very, very big challenges. And if you are here in the future still working within this topic, then you will realize that in Danish hospitals, people are crowded together. There is overcrowding all the time. We have a population that is admitted to hospital quite often. And we talk about 86% of all wards are constantly being full or overfull with patients.
Yes.
While in the Eastern European countries, you are down at 60%. So maybe they kind of swim in antibiotics down there because they don't really care. But there is more room and space between the patients. And I think that is very important for us to recognize, because in Denmark, it's very, very close together. So infection prevention control is sexy because you are so close together.
Yes. I think you made a very, very good point. It's also my impression from traveling in Eastern Europe, especially after the demolition of the wall. I went to Poland for a couple of years, collaborating with Polish colleagues, and it was quite impressive that they had more space, they had cleaner hospitals, but maybe they lacked a little hand hygiene. There was only one wash basin in each department. So, but the problem with space, we still haven't learned it. One thing is that our old hospitals are overcrowded. The problem is the new hospitals we are building will be exactly as overcrowded as the old ones. So they will not improve hygiene to any significant rate, because we move all the problems from the old hospitals to the new hospitals. That's my main concern. That because we have too few rooms, the rooms are too small, everything has to be squeezed into smaller and smaller areas, and that gives rise to infections. maybe I'm just getting old, but old people are also wise people. Yeah.
Yeah, you had a slide where you showed that in the community or in the stocks, you use around, I think, was 100 tons antibiotics, but in the hospital it was only 5 tons. Like this, yeah. So, what can we do to reduce the stock use of antibiotics? Because at the moment you use it because they make money of it.
Well, that's basically not what we have. We have to ask the vets and the farmers to do some solutions on that, because as healthcare personnel, we cannot go into the vet system and interfere. We can say to them, well, we think you have got a problem, but you have to solve it. And the good thing is that these 104 tons are decreasing, little by little every year. Not because of the farmers, but because of the public that demands meat that was from animals raised without antibiotics. So we here can actually interfere and make our influence on this type of system. It's the consumers that hold the solution. The legislators won't. They are too weak. And the industry will not, because they don't care. But the consumers, they hold the key to the solution. That's my conclusion.
And with that, I think we will move on in a very short moment. I know it's getting a little late in the afternoon, so we'll take 5 minutes to refill the coffee for the last half hour-ish part of this lecture series, and then, yeah, see you in 5.
Symposium.
Okay. We will be having the last lecture now on a well-known plant that some people use for medical purposes, other people smoke it, and some people smoke it for medical purposes. And we have Associate Professor Janne Klitgaard from the Institute of Translational Microbiology and biochemistry and molecular biology, and yeah, but you have so many
I
institutes.
put it up there to help you.
Anyway, take it away.
Thank you so much, and thank you to iGEM for inviting me to come here and talk about my really exciting research project. So in Denmark we have this research trial with medical cannabis. But actually, that's not why I'm working with it as a research project. So I actually started more than 3 years ago on this subject. So I'm not just doing this because it's a hype at the moment. I'm actually doing because it was actually found for scientific reasons. So the point of this is actually to try to give you a solution, or one possible solution to all this problem there is with antibiotic resistance. So what do we do when we encounter this problem with antibiotic resistance? We try to develop new antibiotics. And I'll kind of continue in what Hans-Jørgen talked about before about MRSA, because one very efficient antibiotic we have to treat MRSA is vancomycin. And we're also seeing resistance developing against vancomycin, not in Denmark, but in a lot of other countries. So we see these Visa strains, which are vancomycin intermediate Staphylococcus aureus, and also the resistant version. So they are out there, and when we get this kind of resistance, we can't treat the patients anymore. There are, of course, antibiotics in the pipeline. There are companies trying to develop new antibiotics, but it's really difficult at the time. And the few ones that come out, they are not better than vancomycin. And again, we see this really fast development in resistance also towards these new compounds that are actually put on the market. So if we look at what actually happens when we put in a new antibiotic on the market, so if you go down back into the golden age of discovering antibiotics, there was a lot of new antibiotics put out in the market, and the blue bar, when the bar is blue, that means that you can use this antibiotic, it works in the patients, but when it becomes red, that's when you see the antibiotic resistance. So as you can see, for many cases, it happens really fast. You have here the penicillin. It took only 5 to 10 years before we saw the resistance. For some, it takes a longer time, for example, vancomycin. But that's also because it's a drug that we kind of keep in the drawer. We try not to use it because we know it's the last resort we have. And then we have this really big gap. We call it an intervention gap. That is not because people didn't try to make new drugs. They just didn't develop antibiotics. At that time, there was a lot of these lifestyle-associated diseases, diabetes, heart diseases, all kind of cancer came out. So there's so much else to try to work on and solve. And perhaps there was a little bit more money in trying to cure cancer than it was to try to cure an infection. So that means that, well, research go where the money is. So that's why we ended up with this really big innovation gap. And then when we kind of entered the new thousand, or the millennium, we saw that a few drugs was put on the market, but as you can also see, both for linezolid and daptomycin, we actually saw a resistance development very, very fast. So we are in this case where we don't have really good results. So what do we do? Well, the classical targets, if you want to develop new antibiotics, you have been taught this when you went to the university, that we target the cell envelope, we target the ribosome, transcription, or DNA replication. That's the classical ones, that's the compounds you know already. But there are also other opportunities. You can go for new targets, you can try and prevent the cell from dividing, you could target riboswitches, proteases, You could see if you can some kind of interfere with the metabolic pathways. You could also go for virulence. What if the bacteria is there but it's not virulent anymore? Then it kind of doesn't matter. You can also try to prevent it from adhering. Then you could stop biofilm development. What if you can stop proteins secreted? Could be toxins. We could look at quorum sensing, biofilm formation, and so on and so on. So it's actually a lot of new targets in there in the bacteria that we can actually go for. But it takes money, and it takes some people or some companies that are willing to go into this area. There's also another solution, and that is what I call helper compounds. You could also call them adjuvants. You can call them anti-resistant breakers. They have a lot of cute names. So what is a helper compound? A helper compound is what I call an alternative solution to treatment of infections. So it's a drug that has an antimicrobial effect, but it has been approved for other purposes. It could be treatment of schizophrenia, it could be an anti-inflammatory drug, it could be so much more. But if you actually go and look into the effects of this drug, it could also be antibacterial. So it could have this direct antibacterial effect, but it could also be a synergistic effect together with an antibiotic. So that's a new way of looking in how can we actually deal with this resistance problem. So if we can just take, for example, penicillin and give it back to the patients, but just adding another compound, and then we can actually solve the resistance problems. So that is what my research project is about. And what is really obvious is that, of course, it has something to do with medical cannabis. So in the medical cannabis plant, there is a compound called CBD, cannabidiol. And well, there's a lot to say about research in medical cannabis. And as a researcher, I really need evidence. And this is a figure I found on the internet, and it says that CBD has antibacterial effects and that it slowed down bacterial growth. But I have also done the studies, the literature studies, of trying to find the data behind this, and I've only found 3 papers stating that CBD has antibacterial effects, and it's not really the best studies. So of course, we wanted to show if this was really the fact. So we are working on MRSA, because it's my favorite bug. So we took 3 different cannabinoids and tested it against the MRSA strain. So we measured the MIC, which is the minimal inhibitory concentration. It gives you an idea of the lowest concentration that can inhibit the growth of the bacteria. And as you can see, we are actually kind of in a lower range. It's actually quite efficient in itself. And if you compare it to other antibiotics that we use to treat MRSA or Staphylococcus infections, this is really efficient against the strain, and this is really resistant. So it's in the better end. So our thought was, okay, let's take the worst antibiotic we have and combine it with one of these cannabinoids. And what did we actually see? So if we have the MRSA, we also looked at MRSE, we had Enterococcus faecalis, Listeria monstogenes, and in all 4 cases, we could reduce the MIC of the bacitracin, the antibiotics, into almost treatable range for the patients. What we did, also relating to the question, what can we actually do as students at the university, we took a patent on this observation, and we are working on that right now, trying also to get money to continue our research. So we're quite happy that it actually works when we look at it in the bacteria, but we are also scientifics. We want to know, how does this actually work? So I know you all know what a bacteria looks like. So just to say that when we look, in this case, the cell wall of this gram-positive bacteria is really interesting when you have the peptidoglycan, we have the membrane, just to recall on that one. So if we want to look how it works, we took microscopy pictures using an electron microscopy just to see what does the bacteria actually look like. So here we have a Staph aureus, looks very, very nice, this cockeye shape, and it has here its two septum, it's starting to divide, looks very, very nice. If we add the bacitracin, the antibiotic, it looks really nice, almost the same, it's also starting to divide, everything perfect. If we take the cannabidiol and just add that to the bacteria as well, it looks perfect, normal, no problem. But if we combine these two, we end up with this bug, and that looks really, really sick. So maybe you can see, I hope you can see it from the back, but the membrane here is really fluffy. It has several membranes here where it's supposed to make its septum and divide the cells. So we can actually see that the cells stop dividing and this cell is starting to die. So it interferes, it seems, with the membrane of the bacteria. So we try to see what happens to the membrane, and you can measure the membrane potential, and it gives you an idea of the difference in the electric potential between the interior and the exterior of the cell. So you measure this kind of a red-green ratio using fluorescence, and here we have a control. It's an untreated control. That means that here we have a membrane potential. Then you have a negative control where you have a 0. And when we add CBD, you actually see that adding CBD disrupts the membrane potential. And when you disrupt the membrane potential of a cell, it dies. So we can now say that there is something wrong with the cell membrane. But there was also all the other things. So we also wanted to see what happens then with the cell. What happens with the gene expressions. So we did RNA-seq, a global RNA-seq, when we exposed the bacteria to either CBD, bacitracin, or the combination of the two. And what we actually found was a lot of genes that was upregulated or downregulated, but they all had something to do with cell wall. It was an efflux pump, it was a transporter, something again with cell wall, a transporter. So it's all located around the membrane or the cell wall. So again, we have something that says that the effect or the mechanism of this combination is related to the membrane or the cell wall. So on that note, we looked at the cell wall. You can actually peel off the cell wall of a bacteria and analyze it by mass spectrometry and see what is the composition of this cell wall. And when we did that, it was completely normal. There was no change when we added either CBD or the combination. So we are back now in thinking that something happens at the membrane stage that kills the bacteria. So that is what we are working on at the moment. We have made a 3-year collaboration with one of these new medical cannabis companies in Denmark called Antibiotic. They have paid for a PhD scholarship, or co-financed it together with SDU. And in these 3 years, we're going to be more exact on what is the mechanism of action. We're going to do some toxicity evaluation. And the final part of the PhD is going to be on developing a product for topical application, and then do animal experiments to get kind of the first proof that this could also at some point end out with the patients. And then I have to say thank you to Claes. He did all the work, and we have funding from SDU Research and Innovation Organization and also from Antibiotic. So thank you for your attention. I'll take any questions.
My first question is, why did you start looking at CBD oil? How did you identify that was a good idea?
It's a long story, but I have been giving talks on previous projects where I had combining different compounds, and then I was found by a company placed in Jutland, but they were working on extracting CBD from the plant, and they have heard that I talked about combining drugs, and they said, hey, would you please test if their CBD was antimicrobial, because they also found these 2 or 3 papers that was out there, and they just wanted to see if it was true, and then we just tried to combine it, and it worked better than any of the other combinations we had tried before. So this is the most promising helper compound that I've been working with so far. So sometimes it's coincidence what you actually just start up working with in the lab.
Okay, that was it for today. We want to thank all our speakers for giving their time, and we have a gift for them, which is not something that is infectious. It is not a communicable disease, but we have some wine for them. So one for you, and then we have for Ude and Hans-Jørgen, and Alexandra unfortunately had to leave because of a train. So thank you very much to the speakers, and thank you for listening in and joining us today.